So my name's Craig Sten. I work in the Northeast Branch Ecosystems and Threatened Species team for the Department of Planning and Environment. Today I'm coming to you from Gumbaynggirr country, and I just want to acknowledge and pay my respects to traditional owners of this country. So I was going to just give a quick overview of Myrtle Rust in Australia. I think it's probably something that most people listening in today are fairly familiar with. I wanted to talk about some of the impacts we're seeing on the east coast of Australia, both to species and communities, and then run through some case studies for two species I've been working on closely. And also a bit of an extra handful of slides at the end, just giving a bit of an update on that recent detection on Lord Howe Island, because I know it's something that a lot of people are interested in. So Myrtle Rust was first detected in Australia in 2010. It's now widely established on the east coast, but there are several areas that have managed to remain Myrtle Rust free. So southwest Western Australia, Lord Howe Island are two examples. So that means in Australia, the response is not just managing impacts from Myrtle Rust. In some of those locations, it's still about biosecurity and early detection and containment. So in Australia, we have 382 of the known host species. So that's 17% of our known Myrtaceae and 80% of all known global hosts. So we have a very large number of host species. Of the known Australian hosts, 43 are thought to be severely affected. And 5 species so far have been listed as critically endangered due to declines from Myrtle Rust. So they're the two species I'll be looking at today: Rhodomyrtus psidioides, Rhodomyrtus rubescens, but also Rhodomyrtus maideniana, which has recently been listed and is a species I also work on. Lenwebbia species main range, which is on the border between Queensland and New South Wales, and also Lenwebbia species blackall range, which is a southeast Queensland species. So those listed species are only a small part of the total number of species that are impacted by Myrtle Rust. Recently, there was a paper published in Queensland identifying another 16 species that are on extinction trajectories, and I believe Rod Fensham gave a presentation to this webinar series recently about this. And there are several other species in New South Wales that we're seeing decline either across their entire range or a substantial part of it. So I'll quickly mention some of those species now. One of the ones we've been working on recently is the scaly myrtle. In New South Wales, its main distribution is north of Ballina, but it also does occur down as far as Bellingen and Dorrigo. It extends into Queensland, and it also occurs up in northeast Queensland as well. We've also got the silky myrtle, the Dexspermum humile. It again has a fairly widespread distribution in New South Wales, so north from Gosford up into Queensland as far as Maryborough, but then again up in North Queensland between Townsville and Cape York. And finally, the other one I've been looking at recently is Rhodomyrtus beckleri, rose myrtle. It's very widespread. It's quite common in New South Wales. It also occurs up in Queensland as far as Gympie in southeast Queensland, but then again in North Queensland between Mackay and Cooktown. So the species I've just mentioned, most of those and most of the other highly susceptible species occur in wet sclerophyll and rainforest communities, but there are other communities as well that are being impacted by Myrtle Rust in Australia. So most notably the coastal swamp sclerophyll forest, which is dominated by Melaleuca quinquenervia, and also the coastal heath, which is dominated by Melaleuca nodosa, as well as some leptospermum species. And those communities are particularly affected following fire. We have a high number of copper shoots, epicormic growth, and they just continually get hit by Myrtle Rust when they're trying to recover. So today I'll be mainly focusing on the rainforest impacts and wet sclerophyll community impacts. So there are several sites that I've been working on in northeast New South Wales, and Jeff Pegg and others have been working on in southeast Queensland, where we're starting to see multiple susceptible species decline, and these species aren't yet listed. Sometimes they're not even affected across their entire range, but at particular sites we're getting these multi-species impacts. And on the screen at the moment, to the left, you can see a lot of small stems which are a mid-storey of Rhodomyrtus beckleri, and that's totally been removed from this site. On the right, you can also see a lot of dieback, multiple different species, again, all defoliated from Myrtle Rust, and several of those have also died. So these sites and similar sites in New South Wales are just gradually being transformed from myrtle-dominated mid-stories to non-susceptible or non-Myrtaceae species being dominant. And it's something that in New South Wales, at least, we're only just starting to get a handle on. So just another couple images. Again, these are from southeast Queensland. We've got a large Syzygium coronanthem on the left, potentially, a couple hundred years old. In that image, you can see it's mostly defoliated. As far as I know, that tree is now has died. On the right, again, a mid-storey full of susceptible Myrtaceae that have just been removed. So all of those floral resources, all of those fruit resources are just being removed from some of these communities, and often they're being replaced with non-Myrtaceae. So a really good paper on that is the one that Jeff Pegg and others put out in 2017, which is mentioned on the screen here as well. So the species I've been working on mostly are the scrub turpentine and the native guava, so Rhodamnia rubescens and Rhodomyrtus psidioides. So these were both really widespread common species. For scrub turpentine, it occurs from the south coast below Sydney all the way up into southeast Queensland, and across that range it was very common, widespread, occurs in lots of different plant communities, and was also used a lot in revegetation work because it was such a hardy, fast-growing species. It's now been listed as critically endangered in New South Wales, Queensland, and at a Commonwealth level. Rhodomyrtus psidioides is a very similar situation. It's distributed from just north of Sydney, from Gosford up into southeast Queensland. It's more of a littoral rainforest species, but also does occur further inland. Again, it was once very common, and it's been hit particularly badly by Myrtle Rust. So I've been working on both of these species now since they were listed in New South Wales. So one thing that's common to both of those species, and to all of the species that are actually listed as critically endangered, is that they're suffering these rapid declines from Myrtle Rust. Most of the adult plants for Rhodomyrtus, and on some sites for Rhodamnia, had died within the first few years of Myrtle Rust arriving in Australia. We also know that flowering and seed production is affected by Myrtle Rust infection on these plants, and seedling recruitment is basically non-existent. So we know that the plants we're seeing in the wild now are probably the last natural recruits for these species, and they're likely to disappear within the next 5 to 15 years. It's uncertain how long they'll last. So because the rate of decline has been so rapid, and we're losing individuals, we're losing entire populations, so we're also know that we're losing genetic variation within those species, and that genetic variation is essential for long-term recovery, as well as having that adaptive potential for the future, for any changes that may come. So the emergency response in New South Wales has really focused on trying to capture that genetic variation before it's lost, and that's largely been done through germplasm and genetic sampling. So over the past 3 years, we've been undertaking rapid field assessments of rust impacts for these species. And the main aim of that was to try and determine whether or not the impact we're seeing at one site is replicated across its entire distribution. And we've found for the species we've been working on that is the case. There are no areas or populations that seem to be faring better than others. We've also collected leaf samples from all of those sites, and we've been working with the Botanic Gardens down in Sydney to undertake some genomic studies to help inform our ex situ collections. We're also obviously collecting germplasm while we're out in the field, and that's either through cutting collections for Rhodomyrtus rubescens or for Rhodomyrtus psidioides. Sometimes we're also able to just excavate entire suckers, and they're easier to establish in the nursery. So I'll just run through some of these areas a little bit more. With our field assessments, we've found for Rhodomyrtus rubescens that most of the plants we're finding now are quite small. So the graph I've got on the screen here is a height class histogram, and you can see that most of the individuals we're seeing are between that 2 and 6 metre high height range. And prior to Myrtle Rust arriving, we would have found most plants would have been larger trees, 15 to 20 metres high for this species at least. So we do still find some of those larger plants, but they're becoming increasingly uncommon. For Rhodomyrtus rubescens, we also do still find plants that look relatively healthy. So a good level of leaf cover. Sometimes when you first see them, they don't look like they've been impacted much. But we know from other observations that these plants, if they're flowering and setting fruit, they're often not containing any viable seed. So Rhodomyrtus psidioides is a different story again. Most of the adults have died in this species, and what we're finding now is mainly just small sucker fields that are coming up off the roots of those previous adult plants. So in the assessments I've done for this species, most of the plants we're finding now are small suckers. The majority are below 50 centimetres tall, and nearly all of them are below a metre. And when people see these, they often think they're seedlings, but when you excavate down, you can see they're just coming off the roots of the original adult plant. So the largest and healthiest plant I've ever seen for this species was one individual that was about 8 metres tall. It had a very healthy cover of leaves, no sign of Myrtle Rust infection when we saw it, and we're really hoping that this might be a potentially resistant individual. Unfortunately, we've gone back to that site recently and it's been heavily infected with Myrtle Rust, so it can be really hard to know impacts when you're looking at a species just at one point in time. So yeah, it's quite disappointing to see that. Any of the other larger plants we've found have often been in very poor health. So I mentioned that we've been working closely with the Botanic Gardens in Sydney, who've done a lot of their genetic work on this species, as well as a lot of other species we've worked on, and they've been really essential in us progressing a lot of this work. So on the screen at the moment, I've just got a summary of some of that genetic, of the results of those genetic studies. So the two boxes have a couple of maps for each of those species. On the left, we have a map that shows the collection points. So for Rhodomyrtus rubescens, we included 281 samples. For Rhodomyrtus psidioides, 191 samples were included. You can see across that map there, they're color-coded based on latitude. And what we found in terms of genetic variability was that there is variation in those populations, and it occurs across that latitudinal gradient. The map you can see on the right for both of those species shows those same points, but this time the blue points are collections that we have in the Botanic Gardens down in Sydney. So we've got a very good geographic spread of those collections, and the genetic data also tells us that we have good coverage of that genetic variability. So our future priorities for these two species, while we're not doing any widespread germplasm collection anymore, we're still looking for rust-resistant plants. We know with Rhodomyrtus rubescens that they're likely to be out there. We've found them before. For Rhodomyrtus psidioides, it's really uncertain, but you know, just looking in places we haven't already looked and just trying to have a bit more of a complete survey for that species, just to see if there are any sites or individuals that are coping with myrtle rust better. Because we have large collections too down at the Mount Annan Botanic Gardens in Sydney, we're also looking at trying to disperse some of those collections. So trying to manage the risk of having the collection at one location, and also trying to manage the, you know, just the time and effort that goes into managing a large collection. So dispersing that out as much as we can. And we've been working with the Australian Network for Plant Conservation over the last 12 months or so on our Rhodomyrtus psidioides project, and those collections have now been dispersed to at least 5 botanic gardens in New South Wales, ACT and Victoria. And then I think in the longer term, and something we're really wanting to progress over the next 12 months to 2 years, is looking at resistance within these species in a bit more detail. So trying to determine whether or not there is variable resistance through doing screening and myrtle rust assays. Some initial small scale trials for Rhodomyrtus rubescens have found that there are highly resistant plants within that species. Also, that resistance has been observed in the progeny of those plants, so that we know that those traits are being passed on. So in the longer term, our focus is to try and look at whether or not we can use tree breeding approaches to try and transfer some of those rust-resistant traits into locally adapted populations for eventual reintroduction. It's a long-term goal, obviously, and there are a lot of hurdles to get from here to there, but we know it's been done elsewhere. We think it's technically feasible, and a lot of our efforts in the forward couple years will be focused on trying to progress that work. Before I wrap up on this section and cover some of the Lord Howe Island work that I've been doing recently, I just wanted to point to some really good resources that have come out recently in Australia. One is the Myrtle Rust Silent Killer film, which was done through a partnership with Queensland Government, as well as a range of NGOs in Queensland and New South Wales. So that's available on the Australian Network for Plant Conservation page. The QR code on the screen there will take you to the Myrtle Rust page. Otherwise, the web address is at the bottom of the screen. There's also links on that page to the dispersal project for native guava, the Safe Custody of Native Guava project. So you can look at what's been happening and any resources that have been produced around that project. Yeah, so as was mentioned at the start of the webinar, the reason this webinar had to be delayed for a couple of weeks was that I was on Lord Howe Island helping with the Myrtle Rust response over there. So probably half of my work is focused on Myrtle Rust projects on the north coast of New South Wales, and the other half is focused on threatened flora projects on Lord Howe Island, as well as critically endangered land snails on Lord Howe. So I just thought I'd run through what's been happening. It's very early days in the response over there, but I think there's a bit of interest in what's happened on Lord Howe in the past. So Myrtle Rust was detected on Lord Howe back in 2016. The board jumped onto it straight away. They had prepared for a likely outbreak on Lord Howe, and they implemented their control strategy, and they were able to rapidly contain that initial detection and control the outbreak. And Myrtle Rust was eradicated from Lord Howe Island. And as far as I know, it's the only successful eradication of Myrtle Rust ever. Unfortunately though, earlier this year, on the 3rd of February, Myrtle Rust was rediscovered on Lord Howe. And to date, infections have been found on 4 properties, with the most recent infection or active infection found on the 15th of March. So just a bit of context on Lord Howe. It's 600 k's east of Coffs Harbour. It's about 1,400 hectares. It's a very rugged island in terms of its topography. It's dominated by these two large mountains to the south of the island, Mount Lidgebird and Mount Gower. The settlement area, which is where the infections have been found so far, is approximately 350 hectares, has just under 400 residents and about 400 tourists maximum at any point in time. So another bit of context that I think is really important to mention is there's a real history of eradication work on Lord Howe. So pigs were eradicated in 79, cats in 81, feral goats in 99. The largest and most complex eradication on the island was the rodent eradication, which occurred in 2019, and it's actually due for its success check in a few months' time. And also there's been 6 weed species eradicated. So I think that's important context because there's a real eradication mindset when something's discovered on the island. If it's non-native and it's likely to have an impact, eradication is definitely their first response. So there are 5 endemic Myrtaceae on Lord Howe. We've got a large Syzygium fuligari, which is the scaly bark. Impressive, tall, buttressed plants and quite common in the southern mountains. There are also 2 Metrosideros, a highland Metrosideros as well as a lowland one. And then the mountain tea tree, the Leptospermum, which occurs on the mountaintops in the cloud forest predominantly, and Melaleuca howiana, which is a small tea tree that occurs in the settlement area as well as some of the exposed areas. From what we know, the first 3 are likely to be moderately or potentially severely susceptible, and that's based on the fact that they're the only ones that have actually picked up Myrtle Rust infections in the wild. The others are, well, Melaleuca howiana is known from susceptibility trials only. So we're quite concerned about the potential impact on those, and they're really quite common in the southern mountains areas and are also a major part of some of the critically endangered ecological communities on the island, including the gnarled mossy cloud forest. So after the initial detection in early Feb, the board implemented a really rapid containment, control and eradication strategy. The initial plants were controlled with fungicides straight away. All Myrtaceae in the settlement area have been mapped, and that's more than 4,000 individual plants. Majority of those are exotic species to the island, so the Pahudakawa, Metrosideros excelsa, as well as a range of callistemons from the mainland of Australia. So they developed an app on day 1 and they have mapped all of those plants and now they regularly inspect those. Also, a lot of the exotic hosts were removed. So when an infected plant was found, they go through a process of 3 rounds of fungicides and then they follow up with a stem injection to remove the plant. If it's small, they would just bag it as soon as they find it. But on a lot of those properties, they're also removing a lot of the other exotic hosts to try and remove that potential source of host material in the future. So the settlement is obviously just a small area of the island. The southern mountains and the northern hills are all contained in what's called the Permanent Park Preserve, which is like their equivalent to a national park, I suppose. Searches in the PPP began in mid-March. And because of the extensive areas and the really difficult terrain, searches in these areas were prioritised based on the assets that were potentially at risk. So that's susceptible species and at-risk plant communities such as the cloud forest. So those searches have taken place. All the tracks have been searched now. Some off-track areas are starting to be searched, and to date, no Myrtle Rust has been found in the park area as well. So while it's quite early days at the moment, it's too early to know if the eradication will be successful, but the trends are really positive. infections have been limited to a small number of plants, and more importantly, the spore load has been kept really low. And for some plants and some sites, only a handful of branches or leaves were found infected. So getting onto it and containing that infection so quickly, I think, has set them up to hopefully be on that eradication trajectory for the work over there. So those regular searches are continuing, and that includes sweeps of the entire PPP as well and the settlement area, and that's happening every 2 weeks. Follow-up inspections on the known infection sites are happening more regularly, and any follow-up treatments or stem injecting is also occurring. So yeah, I'd just like to acknowledge, you know, the Lord Howe Island board over there have really jumped onto this with everything they've got, and the signs are looking very positive at the moment, but we won't know for sure for several more months. So look, thanks for listening. I hope that was informative, and yeah, thanks for the invite to the webinar series.
Thanks, Craig. That was definitely a very sobering report on what's happening in Australia, but also some good news so far about Lord Howe. We'll keep our fingers crossed. So I guess one of my questions was that how long do some of the species that are under decline, how long are they keeping their leaves on for?
So that seems to be fairly variable. So for Rhodomyrtus rubescens, it can, you know, there are lots of plants out there that look quite healthy at the moment that have a really healthy cover of leaves. I think sometimes it's dependent on when the flush occurred. The spore load, the East Coast this year has had a very heavy spore load. I've heard that similar things are happening in New Zealand. So yeah, it can be variable. Some years they can be quite defoliated and heavily infected. Other years they can look quite healthy.
Sounds good. There are some lots of compliments coming on about your presentation. A great presentation.
Oh, I'm sorry, I've lost your audio there for a sec.
It just lots of comments are coming on about what a great presentation. Some people want to know whether you are going to the Australasian Myrtle Rust conference in June. So to question you in person.
Yep. So yeah, I will be there, and I think I'll be involved in one of the field trips as well, which is a site that has had Rhodomyrtus rubescens on it.
Excellent.
Looking forward to that.
One of the questions is, thanks for a great talk. I was wondering how or if citizen science apps such as iNaturalist have been used in your work to find new individuals or populations of these species.
Yeah, that's definitely been really important for us. In New South Wales, we have our official record system, which is called Bionet. But to get an entry into that, there's quite a process. A lot of other data needs to be provided with the record. So using iNaturalist or ALA, Atlas of Living Australia, which iNaturalist feeds into, has been really important in finding sites where we know recently at least that healthy plants have been observed. So the fact that people often put photos with our iNaturalist records, it's been invaluable. It's been really important.
So another question that I had was just when you were mentioning the different areas that you're monitoring, how often are you going back to follow up with the monitoring?
So for a lot of the sites, initially it was about just understanding whether or not the impact was uniform. So some sites we only intended to monitor once. At other locations, particularly sites where we observed healthy plants, we have remonitored those sites just to see if that was you know, just because it was a particularly good year with low Myrtle Rust spore loads, or whether or not it was true resistance. So it depends. We've changed our sort of return visit time depending on the location and the species and what we've observed. We're just about to implement a project that looks at more plant community impacts, and we're currently looking at a lot of sites on the north coast where we have plot data that predates Myrtle Rust, and we're hoping to revisit those sites where there's a high proportion of susceptible species, and actually trying to document what change has occurred over that period with Myrtle Rust. So that project should be starting in the next 6 to 12 months.
Really good. Right. A question came in regarding the 2 species that you spoke of on New South Wales. Do these 3 species have fauna that relies solely on them for their survival?
I suppose we don't really know that. I would assume that most of the species that either rely on the floral resources or the fruit resources are more generalists. There are lots of different myrtles that also have that heavy flowering and the fleshy fruits that, say, Rhodomyrtus rubescens has. So yeah, unfortunately we just don't really know, and it's something we would have liked to have known more about before this happened. But you know, as with most things, our knowledge of those particulars are often lacking.
That's definitely something that happens in other countries as well. So we realize how important some of these species are when they're threatened with extinction. I think another question is, there's one question about how likely is Myrtle Rust to mutate. I mean, that is always the risk, I think, with the new pathogen, just to see, like, you know, what happens with it, and whether it's going to jump onto other hosts and things. So would you like to comment on that?
Yeah, look, that's probably outside of my area of expertise, but it is something we consider when we're doing a lot of our work. Even though, you know, Myrtle Rust is across the East Coast, I think hygiene protocols are still important, because we don't know if it is mutating. We don't know if other strains are potentially there, and they just haven't been confirmed. So I think from my perspective, you know, that's how we can manage it. But yeah, I can't really comment on the technical side of that.
Yep. No, I think that's definitely something that we monitor as well, which is why it's still an unwanted organism, that we don't want new strains arriving and attacking new plants that we didn't want, that weren't being affected in the first place. So with Lord Howe Island, you mentioned that you were going to be doing the sweeps of the island. How long is that going to be continuing on for?
So at the moment, the plan is to continue 2-week sweeps for a set time after the last active infection. I think at the moment they're planning on doing at least 3 full sweeps at that 2-week interval, and then it's still being decided, but I think the idea is to continue regular sweeps after that as an ongoing surveillance sort of tool for Myrtle Rust, but combining it with other biosecurity searches.
Sounds good. It's definitely very encouraging to hear that it has been eradicated once, and sounds like things are under control over there. Certainly, it's very important for some of our offshore islands here in Aotearoa as well. Well, thank you again for an amazing webinar. That was really, really informative. And that's it for today. So a video of today's webinar is going to be made available on the Beyond Myrtle Rust website in the next couple of days. It will also be emailed to everyone who has registered. The next webinar is on Wednesday, April 19th, from Michael Watts. Michael is a principal scientist with Scion, who specializes in data and geospatial intelligence. His talk is titled Pre-Visual and Early Detection of Myrtle Rust on Rose Apple Using Hyperspectral Measurements and Thermal Imagery. And it's going to be very interesting to hear about that. So thanks everyone. Hi, Rara. See you next time.